So hello everybody to the second presentation of this day. Again, my name is Philippe Pascal and I will moderate this session. The next topic we will be talking about is the biological evaluation plan. This is newly introduced in the 2018 version of 1093-1 and should help you to understand whether and what kind of testing you have to conduct for your biological evaluation, and it should basically put you away from the tick box approach and put more a systematic approach into your biological evaluation. Sophie Michel will be talking about this topic. Sophie has a PhD in biomedical engineering, which she received from the University of Maastricht in 2014. Thanks to her broad experience in chemistry and biocompatibility, she joined the team of study directors at Nelson Lab Europe in 2015, where she currently focuses on the biological evaluation of medical devices through classical biocompatibility tests and chemical characterization. In 2019, Sophie Michel became a member of the ISO 10993 committee. So please, Michel, Sophie, you can start your presentation. Thank you.
Good morning, everyone, and thank you for joining this presentation this morning. This morning, I'm going to talk to you about the biological evaluation plan as a crucial first step in the biocompatibility evaluation of a medical device. But first, what is a biological evaluation plan, or a BEP? So a BEP is actually a document in which you are going to outline the strategy that you are going to adopt to evaluate the biological safety of your medical device. It can be either through testing, but it can be, of course, by other means. And so this document should be written before you step into testing, which would be the phase 2, and before you evaluate whether or not your device is safe to the patient in a biological evaluation report, which will be the phase 3 of the biological evaluation of your medical device. Now, if you are working on medical device for quite some time, you're probably wondering why the notified body and the FDA are actually requesting to have a biological evaluation plan in place. And I think that to answer to this question, we need to go back to where we are coming from. So if you're also working for quite some time in this field, then you will directly recognize this biological evaluation chart, which is taken from the previous ISO 10993-1. So it was quite easy to use because you need to classify your medical device, their contact, their time, and then the type of testing that you needed to perform was just coming in front of you. But the problem with this biological evaluation chart is that it did not encourage the sponsor to understand its material, to understand its manufacturing process, but also to understand how a biological endpoint is actually relevant to the risks which are associated with a specific medical device. And so this ISO 10993-1 was revised. It was not revised in title because the title stayed the same, but it was revised in content to align with the expectation of the title, I should say. And what is this title? It is Biological Evaluation of Medical Device, Part 1, Evaluation of and Testing within a Risk Management Process. And what is risk? This is defined in ISO 14971. This is a combination of the probability of occurrence of harm, so in other words, of a biological endpoint, with the severity of that harm. So how severe could be this endpoint to a specific patient? So how does it translate in the new biological evaluation chart? So this is a new one. What you can see directly is that there is much more biological endpoints which are listed in this chart. The first one, of course, is physical and chemical information, but there is also the presence, for example, of acute systemic toxicity, subacute, chronic toxicity. There is carcinogenicity, reproductive developmental toxicity, and degradation. So you can see them listed here. But what is even more striking is that there is inside, just below these endpoints, X and E. X means that it is a prerequisite information which is needed for the risk assessment. So in practice, only physical and chemical information gathering should be performed in a mandatory way, whereas E means that it's an endpoint that needs to be evaluated for in the risk assessment, either through the use, for example, of existing data, testing, or, for example, evaluation of why this endpoint is not necessarily needed for your medical device. And it's also specified that for particular medical devices, there is a possibility that there will be fewer or additional endpoints than the one which are listed here. So we are moving very much away from the checkbox approach, and you need to evaluate your device in a more risk assessment perspective. So in this framework, what is actually a biological evaluation plan? So a biological evaluation plan is a part of a risk management process. It involves the identification of biological hazard, estimation of the associated biological risk, and determination of their acceptability. Why? Because the Annex B.2.2 of ISO 10993-1 specifies that it is emphasized that simply planning to conduct testing against all of the aspects of biocompatibility identified in the biological evaluation chart does not meet the requirement of this document and of ISO 14971. And the same is said in the FDA guidance on ISO 10993. Such a process, so writing a biological evaluation plan, should generally begin with assessment of the device, including the material components, the manufacturing process, and the clinical use of the device. And then considering this information, the potential risk from a biocompatibility perspective should be identified. And this risk will have to be addressed either by testing, but also by other means. So this is a theory of what we are aiming to do in a biological evaluation plan. But of course, now the real question is, how would we do this in practice? So in practice, we would follow the clause 4 of ISO 10993-1, 2018. So we would look at the configuration of your medical device. What is its size, for example? How does each part contact the patient? We would evaluate the raw materials. We would look if there is any historical data. For example, if it's a new version of an existing device, then maybe we can use some data which was gathered on the previous one. What is the impact of the manufacturing process? What is the impact of the packaging process? And we would look if there is any literature which can be used, for example, on the raw materials. And all this will help us to define the testing strategy. So in this presentation, I really wanted to show you how we would build the BEP by taking real life example. So we are going to evaluate how we would build a BEP for a wearable device as a case study. So now you're probably wondering, a wearable device, but this is actually the most easiest kind of medical device to assess. And really, I want you to ask yourself at the end of this presentation, well, is it that easy to assess a wearable device? And this is really the aim of this presentation, is to show you how each device has its own characteristic, which will make it quite complex if we really think about how the user is going to utilize it. So if we have such a device, a wearable device, the first thing we will do is actually understand how it contacts the patient. So if we look at this product, for example, we have two parts which are contacting the patient. The first one would be the tissue fabric, which will come into direct contact with the skin. This is the one that you can see, for example, here on the finger of the patient. And then we will have the digital interface. So for the digital interface, what we would like to directly do is evaluate from which materials it is made of, and if these materials are very common in our daily life and really daily encountered. And why is that so? Because if that is the case, then we can refer to a specific section of ISO 10993-1. I will show you this one here. So this section says that some medical devices used in either sterile or non-sterile environment include components that can come into contact with a user and gloved hands, such as human interface for electronic equipment or housing for electronics and so on. And if this device component can be shown to be made from material in common use for consumer product with a similar nature of contact, no further biological evaluation is needed. So this would be really interesting to already assess the digital interface part and focus much more on the fabric contacting part. Then for the fabric contacting part, we would want to understand how it contacts the patient and especially which tissue it contacts. What does that mean? So here, if you look at it, you can see that there is two types of skin which are encountered. The first one would be normal skin or mature skin. So this skin would be actually really making a natural barrier for the presence of allergens, for example, virus, and also, of course, to a high extent, chemical compounds. And so for this type of skin, there will be a specific risk which will have to be encountered, but which will not be that extensive. This skin will be also encountered more in children and adult population. If your medical device can come into contact with damaged skin, as would be the case, for example, in premature children, that will be a different story. Because if you are dealing with a premature population, then you are in contact with damaged skin, and this skin would be much more permeable to the presence of allergens, virus, but also chemical compounds. And so that will trigger additional risk that we'll have to take into account. So this is also something that we will directly do in a BEP, understand the patient population. And now let's imagine that we are dealing with children or adult population. Here. Then for this medical device, it will be classified as a surface medical device in contact with intact skin for a certain amount of time. But you can see that whichever the amount of time is, will not impact actually the amount of endpoints that we will need to assess for, but will be physical and chemical information, cytotoxicity, sensitization, and irritation. So at this step, we are going to proceed with physical and chemical information as requested by the ISO 10993-1 guideline. So what will we do in this physical and chemical information section? First, we would evaluate if there is any literature data which can be present on the raw materials, especially for the irritation and sensitization endpoints, because then it can help us to already address this risk. Do you have any biocompatibility testing which was performed by the raw material supplier, for example, and that we could also use to address the endpoints? What is the impact of the manufacturing process? So most often when you are dealing with a wearable device, the manufacturing process can be very small. It can be only cutting and putting into shape. So you can see that stepping from the biocompatibility testing performed by the mass raw material supplier to the endpoints required for your final device will be, I would say, a relatively easy exercise. And then what are the packaging materials? Are they already known and well assessed for for surface contacting medical devices? At the end of this, we will ask ourselves if more information is needed, meaning do we need to consider chemical characterization? And for this, we will refer to ISO 10993-18, which says that the extent of characterization required is determined by the invasiveness and duration of clinical exposure in the intended use. So what we have to consider for surface contacting medical device and in contact with intact skin is that the potential of migration of chemical compounds through the skin barrier is actually quite limited. And also, if you do a chemical characterization, it will be very difficult out of this information to really understand what will be the risk to the patient, meaning which chemical compounds are really going to go through the skin barrier. So the information will not be so useful to adequately address patient risk. And so for this type of medical device, we would identify the materials and the processing and use biocompatibility testing or, for example, literature data or previous data acquired to support the safety to the patient. And then at this stage, we will proceed to the biological endpoints. In that case, that would be cytotoxicity, irritation, and sensitization. And we will evaluate if they are required, but also if they are required, how we are going to address these endpoints. So which biological testing are we going to perform? For example, for cytotoxicity, are we going to go through a MEM elution, which will be more qualitative, or for with a MTT XTT test, which will be more quantitative, or if you go for the Japanese market, are we going to do a V79 colony assay? If irritation is still an endpoint of concern, so if testing has to be performed, then we would probably recommend to do a primary skin irritation test, which is going to adequately mimic the patient exposure of the medical device. It can be that it will be either through extract or by direct contact, if your medical device can be directly placed in contact with the animal. That will be also decided on at a later stage in the BEP that I will show next slide. And for the sensitization test, we will also go for the bular test because it is, again, in contact with intact skin, so it will be most appropriate for the clinical exposure of the device. Then, in the next stage, we will define how we are going to perform this testing from a sample preparation perspective. So for the solvent, for example, we would want to use a polar and an apolar solvent, as described in ISO 10993-12. And then for the ratio, we would want to use a surface area because it has preference to the authority. But then we would also describe how we are going to perform the extraction in time and temperature. So you have to be aware that 37 degrees is the only temperature, but it's a temperature which is only accepted for cytotoxicity. Generally, it's done for 24 hours. However, if your medical device is coming in permanent contact with the patient, especially if it's an implant on an external communicating device, then you might need to consider 72 hours extraction time, especially for the FDA. For all the other testing, then we will select 50 degrees for 72 hours, which is the typical expectations of the authority. So this is how we would build a BEP for adult population and children. But now let's imagine that your medical device comes in contact with premature children. This is the second case. So this is a damaged skin. And here we would end up with a different classification because the skin is not mature, it's much more permeable. So it will be a surface contacting device in contact with breach or compromised surface for a specific amount of time. And you can see that, of course, the more your device is going to contact the patient for an extended period of time, the more endpoints we need to evaluate the device for. What is important to see is that if you're dealing with pediatric, I would say especially premature or neonate, then you have another endpoint which is coming into evaluation, which is developmental toxicity, so reproductive developmental toxicity, that you will have to take into account. So there is no E in this biological evaluation chart, but it comes as an endpoint of evaluation for your medical device. So for this kind of patient contact, we would follow the same way. We would also perform a physical and chemical information. However, we have to recognize that it will be more difficult to rely on literature data because we would need to be able to collect literature data for your raw material on a specific patient population, which is premature children, which is in practice very difficult to find. And then we would ask ourselves, is chemical characterization needed? So remember, for the adult population, we came to the conclusion that it was not needed. Here we are dealing with a premature population. This is a very risky population. The skin is also much more permeable, so that means that much more chemicals could be migrating through actually the skin. And so for this kind of population, then we would recommend to perform a chemical characterization followed by a toxicological assessment to adequately characterize patient risk. It's also important to see that this chemical characterization will also help us to support specific endpoints. And this is listed in ISO 10993-1, so that certain biological tests, especially those designed to assess systemic effects, are not justifiable where the presence of leachable chemical compounds have been excluded or where chemicals have a known and acceptable toxicity profile. And these endpoints are all the systemic endpoints: acute, subacute, subchronic, and chronic toxicity, genotoxicity, and carcinogenicity, and for this population, particularly interesting reproductive and developmental toxicity. So that means that for this device, if you perform a chemical characterization followed by a toxicological assessment, and your margin of safety is above 1, then you can also use this data to address these specific endpoints. So in a biological evaluation plan, we will look how we are going to perform in practice this chemical characterization. So which strategy are we going to adopt? Meaning, for example, from which toxicological threshold of concern are we going to start from? Which solvent are we going to use? Which techniques are we going to select? And of course, we would recommend a toxicological evaluation of the detected compounds to be able to visualize the patient risk. Then we will proceed to the selection of the biological testing, just like we made for adults. But you can see here that although for cytotoxicity, it will not change the type of test to be performed, it does change it for irritation and sensitization. Because once again, the skin is much more permeable, then we have to go for a more worst case administration route. So that means that for the irritation test, we would select the intracutaneous irritation test, and for the sensitization, we would select the Klingman maximization test. And at the end of this, we would also define how we are going to proceed with the sample preparation for this testing. And it will be exactly as I described before for adults, concerning the solvents, the ratio to be selected, and the extraction time and temperature. So we developed the BEP for the two types of patient population, adult and neonate, and that means I'm also reaching the conclusion of my presentation. So to conclude, what would we do in a BEP? We would understand the contact, the type and the duration, the patient that the device is going to be used with to have an accurate device categorization. Then we would gather physical and chemical information and define if more information is needed by performing a chemical characterization study. Then if biological testing is needed, then we will select the tests which are applicable and define which test exactly we are going to do and how we are going to do with the test sample which is going to be selected and the extraction conditions which are going to be used. So remember, this biological evaluation plan is a first phase in your biological evaluation program. And you can see that if you have a biological evaluation plan in place, it will make stepping into step 2, which will be the testing phase, quite easy, but also it will make it much more, I would say, much easier to proceed with the biological evaluation report, which will evaluate the safety of your medical device to the patient. And for this, I want to thank you for your attention. And if there is any question, I will be very glad to take them.
Okay, thank you very much for your very interesting presentation. It was especially important to see that the biological evaluation plan is not just a new requirement that adds more work to the biological evaluation people. It actually helps you to do less testing because you think of what's required before you do the testing and you don't think of what's required after you have done the testing already. So let me ask you some question from the audience. And the first one is, do you think acute systemic toxicity is required if we do subacute and chronic toxicity? Or is it enough just to do subacute or chronic?
So the FDA would still expect that you're doing acute systemic toxicity. You could ask yourself indeed why, but it's just looking at a different perspective. So it's different time. It's also I would say different administration dose, for example, because it will depend on the route of administration. But if you perform, for example, subacute and chronic per implantation, then the acute will be by injection. So it's not necessarily the same route of administration. And in practice, the FDA will still request that you're performing acute, even if you have already performed subacute and subchronic.
And in the other European markets?
For the European market, it's a little bit more uncertain. It's always the difficulty with the European markets.
People should speak first if the notified body, right? So let me ask another question we received many times from the audience. The FDA always recommends GPMT sensitization assay, even for intact devices. What is your experience with pilot test? Is it acceptable by the FDA?
So I will think that it is acceptable to the FDA, yes. It's always a question of having a discussion with the FDA. I never know how I would say the sponsor justify this. I think if you have a good justification in place, like I explained, discussing about how would be the type of contact, for example, what will be the skin, I would say, contacting a surface, how mature it will be, then I would think that the Buller test, yes, would be acceptable. But I think that you need to have the proper justification in place to have this, to show to the FDA that it is relevant to your specific medical device. Otherwise, they will always select the worst case route of exposure.
Okay, understood. So let me ask another question. Can the chemistry testing be used to justify no animal testing? And in what cases can you, in what cases can't you?
So the chemical characterization testing, it will not, it will only be, you will be only able to use it for the systemic test, as I showed, so the acute, chronic, and chronic systemic toxicity, the genotoxicity, the carcinogenicity, and the developmental toxicity. That is the only endpoint for which chemistry could replace them. For cytotoxicity, sensitization, irritation, pyrogenicity, implantation, and hemocompatibility, then chemical characterization cannot be used. This is specified in ISO 21726, which is about TTC thresholds. And so for these specific endpoints, you will need to use either preliminary data, or proceed with the testing. But chemistry will not be able to support you.
So it's definitely not a yes or no question. It depends on the
Yeah,
case.
it needs to look. You need to look at the endpoint that will be present for your medical device. You need to look at which endpoints are applicable. But for sure, I think there is a little bit more of freedom on this on the European market. Definitely, the notified body really are looking for less animal testing, so they could accept chemistry in specific case to address irritation and sensitization. For the FDA, it's definitely a no go. So they will not accept it if you go to the Euro American market.
Okay, understood. Thank you. So we have another very important question. We remember 10 years ago, people would always do biocomp testing at time zero. But now we have this new lifetime concept where you have to know whether the biocompatibility changes over time. So how would you address the device lifetime for such a device such as the wearable device you have presented to us?
Yeah. So that's a very tricky question. Huh? Yes, now you are asked to take into account indeed the shelf life of your medical device. And this is, I would say, a very difficult exercise, also because it's very new. So we don't have so much, I would say, experience on how the notified body or the FDA even would look at it. But that for this purpose, we would also look at, I would say, the endpoint and what are the materials. So if the material are, for example, often used, so for a wearable device, I would not be too concerned about the shelf life of the product. So I'm speaking more about the wearable device for adults, once again. For neonates, that could be a different story, for example. For the endpoints which could not be addressed by chemistry, then you could consider cytotoxicity, just performing a cytotox at the end of shelf life, because it's a test which is very sensible. It's quite quick. It's non-expensive. It does not involve animal. So that's a good representation already of the biocompatibility. I would say the biocompatibility of your medical device. But for the longer endpoint, then you might need to consider performing also chemistry, for example, at the end of shelf life, and especially if you're dealing with a polymeric device. So with metallic device, once again, we are not so concerned about the fact that they are going to evolve during the shelf life because they are very stable. But for the polymeric device, it's not the same thing because, for example, you will have antioxidants, you will have stabilizers, and this might decompose among the shelf life of the device. So it's quite tricky um, to say to address this kind of device. I would say from a theoretical perspective. But it's a very difficult question because indeed we do not have experience on how the authority are going to address them at this moment. So it's more a
Yeah,
theoretical
absolutely.
exercise.
Yeah, absolutely. We're trying to address new requirements, and we don't have yet the experience to really know how to address them properly. But thank you very much for your answer on that question.